Before discussing the theories of muscle contraction, let us discuss how myosin and actin work in synergy for muscle contraction. Now remember that interaction occurs in this way. Interaction of one myosin filament, two actin filaments, and calcium ions to cause contraction. In a relaxed muscle, a complex called tropin and tropomyosin covers the on switches on the actin filament, preventing muscle contraction. But when there is a high level of calcium ions, they stop the complex from blocking the switches. We don't fully understand how this happens, but it's believed that when calcium ions attach to a specific part of the complex, it causes the complex to change shape. This change pulls a covering called tropomyosin away from the switches on the actin filament, allowing them to connect with the grabbing hands of the myosin molecules. This connection triggers muscle contraction. It's important to know that this explanation is not certain, but it suggests that calcium ions change how the tropin and tropomyosin complex and actin interact, leading to muscle contraction. Now, to understand the basic mechanism of muscle contraction, two theories are followed: sliding filament theory and walk along the theory. The mechanism of muscle contraction is known as the sliding filament theory, which involves two states, relaxed and contracted. In the relaxed state, the actin filaments barely overlap, and the ends of the filaments do not extend far into the myosin filaments. In the contracted state, the actin filaments are pulled inward, causing them to overlap with the myosin filaments to the maximum extent. The Z discs, which anchor the actin filaments, are also pulled closer to the ends of the myosin filaments. So, what causes the actin filaments to slide inward among the myosin filaments? This is due to the forces generated by the interaction between the cross bridges of the myosin filaments and the actin filaments. Under resting conditions, these forces are inactive. However, when an action potential travels along the muscle fiber, the sarcoplasmic reticulum releases a large amount of calcium ions that surround the muscle fibers rapidly. These calcium ions activate the forces between the myosin and actin filaments, initiating contraction. To enable the contraction process, energy is required. This energy is obtained from high-energy bonds and ATP molecules, which are broken down into adenosine diphosphate, ADP, releasing energy. The next one is the walk-along theory. When the actin filament is activated by calcium ions, the heads of the myosin cross-bridges are attracted to the active sites on the actin filament, starting the process of muscle contraction. One hypothesis to explain this interaction and contraction is called the walk-along or ratchet theory. According to the walk-along theory, the heads of the cross bridges attach to and detach from active sites on the actin filament. When a head attaches, it undergoes changes in internal forces, causing it to tilt towards its own arm and dragging the actin filament along with it. This tilting movement is called the power stroke. After tilting, the head automatically releases from the active site and returns to its original position. Then, it attaches to a new active site further down the actin filament, tilts again, and creates another power stroke, causing the actin filament to move another step. This process continues as the cross-bridge heads bend back and forth, walking along the actin filament and pulling the ends of the actin filaments toward the center of the myosin filament. Each crossbridge operates independently, repeatedly attaching and pulling in a cycle. The more crossbridges in contact with the actin filament at a given time, the stronger the force of contraction. In simpler terms, the heads of the myosin crossbridges attach to the actin filament and pull it along, step by step, like walking. Each crossbridge works independently, repeatedly attaching and pulling to move the actin filament and generate force for muscle contraction. The more crossbridges involved, the stronger the contraction. The whole mechanism of muscle contraction depends on these two theories, and the graph for the contraction can be made like this. Now, in this whole process of muscle contraction, ATP plays a vital role. It is the basic unit of energy for muscle contraction. When a muscle contracts, it needs energy to perform work. This energy is obtained by breaking down ATP into ADP. The more work the muscle does, the more ATP is broken down, which is known as the Finn effect. But what is the Finn effect? Let me tell you that as well. The Finn effect refers to the phenomenon observed during muscle contraction where large amounts of ATP, adenosine triphosphate, are cleaved to form ADP, adenosine diphosphate. The amount of ATP cleaved increases as the muscle performs more work. During muscle contraction, ATP is essential for providing the energy required for the interaction between myosin and actin filaments, allowing the muscle fibers to slide past each other and generate force. As the muscle exerts more force or performs more work, a higher rate of ATP cleavage occurs. This increased ATP cleavage is a result of the increased demand for energy to fuel the contraction process. The Finn effect basically highlights the relationship between the amount of work performed by the muscle and the amount of ATP that is cleaved. It demonstrates the dynamic nature of ATP utilization in muscle contraction, with the ATP molecules being continuously broken down to ADP to release energy for muscle activity. So, let's go through the steps of this process. Before contraction starts, the heads of the crossbridges in the myosin molecules attach to ATP. The ATPase activity in the myosin head quickly breaks down ATP into ADP and a phosphate ion. These products remain bound to the myosin head. At this stage, the myosin head is in a conformation where it extends perpendicularly towards the actin filament but is not yet attached to it. When the troponin and tropomyosin complex binds to calcium ions, it causes the active sites on the actin filament to be exposed. This enables the myosin heads to bind with these active sites, as illustrated here. The attachment between the crossbridge head and the active site of the actin filament triggers a conformational change in the head. This change prompts the head to tilt towards the arm of the crossbridge, initiating the power stroke. The energy required for the power stroke comes from the previously stored energy, much like a cocked spring, due to the conformational change that occurred when ATP was cleaved earlier. By breaking down ATP, the myosin heads are primed with energy and ready to interact with the actin filament, leading to muscle contraction. Once the myosin crossbridge head tilts and initiates the power stroke, the ADP and phosphate ions that were attached to the head are released. At the site where the ADP is released, a fresh molecule of ATP binds to the head. This binding of ATP causes the head to detach from the actin filament. After detachment, the newly bound ATP molecule is cleaved to begin the next cycle. This starts a new power stroke, where the energy stored in the cleaved ATP recocks the head back to its perpendicular position, ready for the next power stroke cycle. When the recocked head, with its stored energy from the cleaved ATP, attaches to a new active site on the actin filament, it becomes uncocked and provides another power stroke. This process continues repeatedly until the actin filaments pull the Z membrane against the ends of the myosin filaments, resulting in muscle contraction. The cycle repeats unless the load on the muscle becomes too great for further pulling to occur. That was all about the muscle contraction mechanism in detail. Now we also need to throw some light on how energy consumption takes place during muscle contraction. But that is the detailed topic for another video. Keep watching.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.